Ladies and gentlemen, colleagues, organizers, good morning and welcome. First of all, I would like to thank the organizers for the interesting and very well done conference. I want to thank you also for the dinner of last night, which was very good, delicious. And being in Rome, the eternal city, was impossible to refuse the invitation, particularly in this period of the city, the year pre-spring, when the city shines in the sun, the air is ascended, and the food is delicious, and the wine is excellent. Just a little introduction about our country and our city. And we are very proud. Okay. Now, this is university. I work in two universities. One is in Chieti and one is in the US, in Boston, Massachusetts, at Tufts University. This is the campus in our university in Chieti, in Italy, in the central Italy, which is a nice campus with the monument, the Cascella monument. The guy, it's a very nice, a very good sculpture, very famous, as you can see there. And unfortunately passed away some years ago. And this is Tufts University, Boston. Actually, this is Medford, which is very big, but medical school is not there. It's in Chinatown, downtown Boston. So this is the campus, very huge campus in Boston, outside Boston. Now, today we're going to talk about some immunity cells. Particularly, we're going to talk about mast cells. We know that mast cells are very important in allergic diseases. And we are going to talk about allergens, inflammation, cytokines, and also we combine some infections, the eye infections, with immunity. Mast cells are immunity cells that everybody knows that the mast cells intervene in allergic diseases, but only specialists knows that mast cells can be mediators of inflammation without the degranulation, which is very, very important. Because in some way, mast cells act as macrophages. When you stimulate, when you trigger mast cells, you have the release of immediately release of histamine and proteases. But later on, mast cells release interferon gamma, tumor necrosis factor. I want to point out this tumor necrosis factor. Tumor necrosis factor is a cytokine. Mast cells, this is the only cell that store tumor necrosis factor, because other cells make tumor necrosis factor through the protein synthesis. So when you activate the mast cells, takes time to produce tumor necrosis factor, while in other cells. But in mast cells, there are two ways to produce tumor necrosis factors. One is immediately with the degranulation, which happens in seconds, immediately. When you have something in the eye and you scratch a little bit, immediately mast cell degranulates, because mast cells are like sentinels in the vessels, and to defend the body against any insult. Mast cells also produce prostaglandins through the activation of in the beginning from phospholipase, and then after that arachidonic acid cascade, produce cytokines like granulocyte colony stimulating factor, which is this is a very interesting compound, very interesting cytokine. Unfortunately, cytokines are very interesting, but the therapy with the cytokine is still under discussion. It's not too many cytokines are available for therapy. Tumor necrosis, granulocyte colony, a little bit of a pointer here. Granulocyte colony stimulating factor, especially granulocyte colony stimulating factor. This is a granulocyte monocyte colony stimulating factor, which is a little bit different. But the granulocyte colony stimulating factor is very, very interesting, because, as you know, when you get in tumor cases, when you get when you have with people that has tumor and has needs chemotherapy and radiotherapy, the immunity system decrease a lot, so the people can die by infections, especially virus, but especially bacteria. But now injecting granulocyte colony stimulating factor the night after the therapy, the radiotherapy or chemotherapy, in the morning, you have immediately replaced the immunity system, so the people, they don't die anymore for this. It's very, very interesting. Why is this going by itself? In addition, activation of mast cells produce tryptase, ICAM, and substance B, which is an inflammatory compound isolated by, in our group in Boston, by Susan Lehman, Nobel Prize candidate, because she discovered also neurotensin in the brain. And the serotonin and leukotrienes, these are all inflammatory compounds. So the cytokines, you can see the cytokine appear here. And this is one of the major cytokines, but there are more cytokines released. This is what's the mast cell appear in the tissue, because I didn't say that mast cell is not a circulatory system, it's in the circulation in the blood. It's a tissue cells. And you see here, which trip and blue staining, and the mast cells are in the tissue inside, and here you can see mast cell in electron microscope done by Letourneau, our colleague in Boston. And you see here also a mast cell. It's a very interesting, the granules, and this mast cells was activated because you can see here the degranulation the degranulation is starting here, and then produce inflammatory mediators immediately in seconds. Mast cell mast cells derive from CD34 and CD38 cells from bone marrow, and they go to maturation in the tissue, as I said. I put this slide because the people that are very interested in this, during the COVID, we are first group to say that the people were killed by cytokine production in the lung, because the cytokines, inflammatory cytokines, are very powerful compounds and induce edema in the lung, so the people can breathe and die. How this happened? You see here the virus, SARS-CoV-2 binding toll-like receptor. Toll-like receptor is, as everybody, as you know, is a receptor, ancestral receptor. And in this case, there are 9 or 11 toll-like receptor. Toll-like receptor 2 and 4 binds the virus and then produce a biological effect inside the mast cell and release IL-33, TNF, IL-1, and IL-6. TNF, IL-1, and IL-6 are the most powerful inflammatory that we know. And there are no clinical therapy for this, unless with the IL-1, because there is an IL-1 receptor antagonist discovered in our group in Boston at Tufts by Charles Di Rarelos, which was my boss. And he discovered IL-1 receptor antagonist. So when you treat the people, for example, with the rheumatoid arthritis, with IL-1 receptor antagonist, you block IL-1 and you get the people that gets better. Here, there is a macrophages. So it means that mast cell and macrophages collaborate each other to inflame the tissue. The macrophages also is activated by toll-like receptors 7 and 8. The activation of a macrophage also produces TNF, IL-6, and IL-1. Here we have an autocrine loop. What does it mean? When the cell produce, when the cell produce, IL-1 receptor antagonist, there was it there. So when the cell produce IL-1 make an autocrine loop, goes back to the cell and continue to stimulate the cells. In fact, there is an interesting article published in Journal of Immunology, IL-1 induce IL-1. And that was one of the big steps. And this is make the increase of inflammation, because IL-1 is done by the activation of mast cells or macrophages, and then IL-1 restimulates. In fact, IL-1 activates also mast cells to produce IL-6. So there is a network, big network, inflammatory network. And maybe this is interesting for the people that study inflammation in the eye. As I said before, the mast cell derives from CD34 and CD38, negative from in the bone marrow. And the bone marrow, in the bone marrow, they became CD34 positive and CD38 positive. And then in the tissue, they go to maturation and became mast cell, mature mast cell. Mast cell produce 5-HT, produce also endocannabinoids, prostaglandin E2, histamine, and TNF, which have receptors in the brain and produce pain and depression. Also, depression may use the release, neurotransmitters may use the release of inflammatory cytokines. I wanted to just say what big interest the people has in cytokines. We, in the time of COVID, we published this paper in a very small journal. Surprisingly, we had 2,115 citations because it was the first paper in the world that said that the people die by producing the cytokine, inflammatory cytokine, and then inducing edema in the lung. So it's correct in this case to give corticosteroids, even though corticosteroids are immunosuppressive. But you don't think about immunosuppressive when the people are dying. You have to save life. So it's correct to give corticosteroids for short time, just to reduce the edema in the lung and make the people to breathe correctly or better. So cytokine storm. Cytokine storm is linked with the production of cytokines. As I said before, one cytokine stimulates another cytokine, one cytokine inhibits another cytokine, but mostly of the cytokines are inflammatory. In our university in Boston, Tufts, Charles Di Narello, a guy originally Italian, it's American, where I had the fortune to work, is a Nobel Prize candidate too, this guy, because he isolated, he purified interleukin-1, and also he made anti-inflammatory cytokines, IL-37 and IL-38, which are anti-inflammatory cytokines, and we expect can be used in therapy in inflammation. And this is one scheme of the inflammation, where for example, in the skin, in the lung cells, when the Langerhans cells are activated by various agents like a cytokine, microbial toxin, microbes, antigens, etc., the cells are activated and immune cells are recruited. Cytokines, when you recruit the immune cells, they make cytokines and chemokines, and then IL-1 and IL-6, which increase inflammation. I don't want to bore you describe this complex slide, but I want to just say that in the eye, there can be viral infection, bacterial infection, fungal infection, parasitic infection, protozoa, and other infections. Each infection can cause, can be induced by specific microbe. For example, a viral infection can be herpes zoster, herpes simplex, and cause different disease. Okay, going back to mast cells, mast cells play a crucial role against eye infection through their ability to recognize pathogens and initiate inflammatory response. Mast cells express PRR, which detect the pathogen associated molecular patterns, PAMs, from microbes, which microbes with linked with binding the toll-like receptor, LPS, viral infection, etc. Another, I wanted to put this slide too because the people here are most high doctors. We have an infection here by bacteria, virus, fungi, and parasites. And this is what's happened. This microorganism stimulates PAMs, NFKB. NFKB is inside the cells. And when you induce NFKB, nuclear factor kappa B, you produce interleukin-1, interleukin-6, interleukin-8, which is a chemokine, and TNF, which cause damage to ocular tissue. In addition, microbes can stimulate innate and acquired immunity. Innate immunity makes the release, the generation of C3A and C5a complement fractions, and this produces cell lysis and therefore inflammation. Acquired immunity activate B cells to produce immunoglobulins, to produce antibodies, IgA, IgG, and IgM, and which make pathogen neutralization, as you know. Here, the mechanism of what's happened when the virus binds the receptor, Asher receptor, as happened in the COVID. The virus binds the receptor here, Asher receptor, and angiotensin converting enzyme stands the acronym Asher receptor, or P2X7R. The virus binds the receptor and activated macrophages. Macrophages activated produce inflammasome and caspase, which are precursor of IL-1. And then the cytokines production after the inflammasome and caspase, there are release of cytokines, secretion of cytokines, which stimulate, which damage the lung alveolar cells, and also cytokines make a cytokine storm. And this is kills the people in the case of the COVID. The virus also induce ROS, nitric oxide, and metalloproteinase. ROS and nitric oxide killing the pathogens and damage to host cells. Metalloproteinase damage to the extracellular matrix, corneal ulcer, produce corneal ulcer and keratitis. Again, mast cells are activated. This is a receptor, toll-like receptor, microorganism acts here, and when mast cells are activated, degranulates, and the degranulation produce histamine, protease, and TNF, which worsening keratitis. VEGF, as the doctor said before, it's produced by mast cells, VEGF, and induce neurovascularization, a worsening of keratitis too, because VEGF is absolutely inflammatory compound. When the microorganism binds the receptor, we have a phosphorylation, and we have a biochemical cascade here, JAK-STAT production, with the release of the generation of IL-4 and IL-13, and which contribute allergic harm. The phosphorylation also induce SYK kinase, triggering a MAP kinase PLK and then KB, and this one is the last step to produce inflammatory cytokines, and inflammatory cytokines makes inflammation and participate in conjunctivitis and keratitis. Now, something about cytokines, just to summarize something about cytokines. Which are cytokines? Cytokines, first of all, I would say cytokines are protein. Protein that are immunoregulators, but also, when it's produced in excess, are powerful pro-inflammatory compounds, which is very, very dangerous for the body. Cytokines are protein secreted by the cell that mediate the immune system and inflammation. This is the first sentence. Cytokines are produced by immune cells in response to microbes and other antigens. Different cytokines stimulate diverse responses of cells involved in immunity and inflammation. Now, there was a little confusion about cytokines. Somebody called monokines, lymphokines, cytokines, interleukins, chemokines. Okay, monokines, you call monokines when the cytokine is produced by monocytes or macrophages. You call the lymphokines when they are produced by lymphocytes. Interleukins, it means between two cells, make biological effect between two cells. Chemokines are chemoattraction, are cytokines that chemoattract the cells in the site of inflammation. But somebody said, now all of them we call cytokines. No more this monokine, some people still call lymphokine, monokines, etc. But most of the people now, they use cytokines for all cytokines and chemokines for the all chemokines, because there are many chemokines. In the last period of the scientific evolution, there was discovered many cytokines, chemokines. Cytokines are pleiotropic and redundant. It means that it can attract the cells, and also one cytokine is redundant. One cytokine can make the same effect of another cytokine, which is redundant. And they also have an autocrine action. As I said before, autocrine. The cell produces one cytokine, and then the cytokine goes back to the cell to continue to induce inflammation. Did you ever ask yourself why? Okay, interleukin-1 is a mediator of the fever. Everybody knows this, made by macrophages. Did you ever ask yourself, if there is an autocrine loop, why the fever after a while goes away by itself? It's a contradiction, because IL-1 is produced, autocrine loop, and circulation, only this will never stop. But why it stops? Because the macrophages later on make IL-1 receptor antagonist. That's what Dinarello discovered. And the IL-1 receptor antagonist binds the IL-1 receptor. So IL-1 cannot stimulate the cells anymore, because there is the place is occupied by IL-1 receptor antagonist. Okay. After a tissue damage, are result the tissue release inflammatory cytokine of IL-1 family induction, including the anti-inflammatory cytokines, are produced. Inhibitor of this family, such as IL-1 receptor antagonist, IL-37 and IL-38, this is the last cytokines was discovered that inhibits the cytokine interleukin-1, can therefore provide pain relief and improvement of inflammation. Now, long time ago, we published in Blood an interesting paper, and they had also this one many citations. When you inject chemokines, for example, Orantes, made by T cells, you inject the Orantes in the rat skin. You see here, this staining here, these are inflammatory places, spots, this inflammatory spot. So here, when you inject the PBS, it doesn't make any inflammation. But when you inject Orantes, this is when you inject LPS, a positive control. LPS, we know that it is an inflammatory product from bacteria. Here, when you inject Orantes, you see you attract the mast cells in this spot in the skin of the rat. And when you inhibit the Orantes, the inflammatory induction by the Orantes. It was very interesting. because he was also one of the first publishing about mast cells and chemokines. Here, more clear. This is all. This is that was a PCR, polymerase chain reaction before. Now here, when you inject under the skin LPS, lipopolysaccharide, here on the right, you see how many immune mast cells arise to try to defend the tissue from the lipopolysaccharide, which is a bacterial compound. Here, when we injected the chemokines also, not like LPS, but also recruits the inflammatory cells. Here we use the inhibitor of the chemokine, and here this is negative control, which is LPS. Histidine decarboxylase is the only enzyme that characterizes the histidine, which is a product of mast cell. Histidine decarboxylase, you see here, in the PCR, this is LPS. It means that LPS induces histidine decarboxylase. Randes here also induce less than LPS to confirm the previous results. Randes inhibits also LPS. Anti-Randes abolish completely the effect of Randes, and this is WW mouse, which are genetically they don't have mast cells, in other words. This is genetically without mast cells, this mouse. In fact, when you use Randes, since they don't have mast cells, you don't have histidine decarboxylase induced. Tell me when I have to stop, because I can go over it until tomorrow. Okay, this is what I said before to Dr. Jackson. I learned this method in London in the beginning of the 80s to induce granuloma with the potassium permanganate in the rat. The spot here you see, white spot, this is when you inject potassium permanganate, you induce chronic inflammation. This is a very interesting method because you can study chronic inflammation. You can measure the cytokines, you can measure protease, you can measure whatever you want with this method. And I have to thank to Dr. Willoughby that, teach me to do that, to use this method. After a week, you get a granuloma, white granuloma full of calcium inside, chronic inflammation. So you have to inject 1 to 40 saturated water solution, 1 to 40. If you want to use this method, it's very interesting for your experiments, for your studies. You saturate the potassium permanganate in the water. After that, you make the dilution 1 to 40 of KNNO4, potassium permanganate, and from this, you inject under the skin of the rat the 20 microliters of 0.2 milliliters. So injecting under the skin, after 1 week, you get this big granuloma, and you can use it for your studies. Okay. Again, when you use Rantes to recruit the mast cells, and then you isolate the granuloma, you're plotting the tissue, and then you're going to measure, you stimulate with the LPS, or you inhibit with the dexamethasone, and you inhibit the inflammation here, while LPS induce inflammation. We already talked about this, cytokines and inflammatory cytokines starting from stem cells. We already said that. There are some cytokines that need to make mature the mast cells, and like IL-3, GM-CSF, and IL-1, and then this produce other cytokines that activate megakaryocytes, macrophage, neutrophils, eosinophils, basophils, and erythroblasts. LPS also act like IL-1, binds toll-like receptor in CD14 macrophages. Cluster differentiation 14 is the receptor of macrophage, and so LPS activate the macrophage caspase, which produce a caspase. Caspase activation is necessary for the production of pro-inflammatory IL-1, and then this one can also switch to produce IL-37. IL-37 can inhibit inflammatory response and also immune response, since IL-37 inhibits the receptor of IL-1. It's and IL-18. Again, this is also the skin. Any trauma or any antigen bindings to mast cells that activate the inflammation, and when you have a trauma or you activate the mast cells, you produce cytokines and then stimulate immune cells. And this one, act the immune cells and the cytokines stimulates the other immune cells to make inflammation. This one. How long? It's finished?
Almost we crossed the time.
Okay. Thank you. Tell me when I have to finish. We published in a very good journal. Was Trends in Immunology. Good reputation. This journal has a good reputation. We published. The paper was the title of the paper was Dr. Jekyll and Mr. Hyde. Mast cells are Dr. Jekyll and Mr. Hyde. Why? Because in some way, when mast cells are activation, it favors the tumor growth, but also makes some proteins that are detrimental for tumor. And here is, this is the products. This is the products that stimulates mast cells and make the tumor growth. You see how many? This is the tumor cells, and this is our mast cells around. Because we ask ourselves why the tumor cell, the tumor tissue is surrounded by mast cells. Mast cells occur in the tumor tissue and increase the cell proliferation. I4 VEGF, for example, VEGF increase the cell proliferation because it make a new vascularization and also increase metastasis. While in the other side, some compounds inhibits the tumor growth, which is here. And you can read, you can see there that IL-6, TNF, interferon gamma, transforming growth factor beta, they inhibit the tumor growth. While histamine, NGF, VEGF, IL-8, which is a chemokine, CXC chemokine, increase the tumor growth. This is too complex. Just a few words on fever. We said that interleukin-1, many years ago, the people, they thought that interleukin-1 was produced by neutrophils. That's not true. Interleukin-1 is produced only from macrophage cells. And macrophage cells are very important inflammatory cells. In fact, interleukin-1 can induce other cytokines, as we said before. Interleukin-1 also stimulates COX-2 enzyme. COX-2 is part of arachidonic acid cascade. COX-2 belongs from phospholipase, phospholipase A2. What is the mechanism? When you have the phospholipids in the cells are organized. When you disrupt the phospholipids produce phospholipase. Phospholipase induce arachidonic acid cascade. When you activate arachidonic acid cascade, you have prostaglandin production, lipoxin A and B production, discovered by my friend at Harvard in Boston. He discovered lipoxin A and B, and also leukotrienes A, B, and C, which are slow-reacting substance of anaphylaxis. These leukotrienes are pro-inflammatory in diseases where the mast cell intervene. This other one, but this is what's happened when the interleukin-1 goes stimulate the cells. There is a cascade, biochemical cascade, leads to cytokine generation and inflammation. This cascade can be inhibited by IL-37, which inhibits MYD88, this enzyme. And this is very interesting. Okay. Another example, when you induce interleukin-1, when you use interleukin-1, you inject the interleukin-1 in the rat pod, you can see here the inflammation evident. Here, when you use anti-interleukin-1 and there is an inhibition, and this is anti-IL-1 plus IL-1, which is less than this, but higher than that. This is PBS. When you stress, don't worry about it, I'm finished. When you stress the rat in the plastic tube, you keep the rat inside the plastic tube for many hours. The rat gets stressed. Then you sacrifice the rat and you look at the brain. The brain is full of mast cells which produce cytokines and inflammation. Yeah, it's long talk. This one, okay. We published in Proceeding National Academy of Science a interesting paper where we show that mast cells are important in several diseases, brain diseases, like autism, for example. Why mast cells are important in autism? Nobody knows why the children has autism. We found that the children, mostly of them, born with mastocytosis in the gut. The mastocytosis can be, for some reason, can be activated. So this high number of mast cells can be activated by the environment. Since the blood-brain barrier is not formed yet, because of children, young children, they born normally, the autistic people, the children. The blood-brain barrier is not formed yet. So the mast cell make cytokine, inflammatory cytokines. The blood-brain barrier is not formed, so the cytokines pass the blood-brain barrier and damage the brain, and they became autistic. This is my boss Theoharides in Boston, and this is Susan Lehman, the woman, very smart woman, unfortunately now sick. She isolated substance P and discovered neurotensin, sorry. Many thanks to my group, Theoharides, Susan Lehman, and all these people came from all parts of the world and attached in my lab. Thank you so much for your attention.